Thank you all so much for being here for this webinar, and a huge thank you to Portable Photosynthesis Systems for having me. Like Carrie mentioned, my name is Elizabeth Forbes, and I'm a postdoctoral researcher at Yale University School of the Environment. I'm also associated with the biology department at Memorial University in Newfoundland, Canada. And today I'm going to be talking about zoogeochemistry, and I will define that term, and also how to innovate technologies to measure ecosystem carbon cycling dynamics at the scale of the way that animals interact with their ecosystems. So just a few quick notes before I get started. I'm going to be sharing a ton of photos from fieldwork today. So unless otherwise indicated, the photos and the data that I'm showing to you either come from myself and my colleagues. And I'm going to acknowledge my colleagues in this work as much as possible. I am just beyond privileged to get to travel to new to me places like the fjords of Newfoundland, Canada, and as you'll see in a few minutes, the savannas of Kenya, among other places. And I want to acknowledge the gratitude that I have for the universities, the institutions, and the people that have facilitated all that research. And I especially want to point out that a lot of the work that you'll see today took place on colonized lands that displaced or was unceded by Indigenous communities, like the Mi'kmaq First Nations tribe in Canada, and many Indigenous communities in central Kenya, including the Maasai, the Kikuyu, the Samburu, and more. So I'm going to walk through a few topics today and a few case studies. And first, we'll discuss what zoogeochemistry is. I promised I would define it. And then we'll move on to a case study of zoogeochemistry in action in central Kenya. Next, we'll move on to the development of zoogeochemistry-specific tools for monitoring soil carbon dynamics. And I'll wrap up with some discussion of my current research where I'm applying all of those previous topics to my study of the moose effects on carbon cycling in the boreal forest. So first, what is zoogeochemistry? This is a bit of a trick question, as myself and a network of colleagues, some of whom are here, are currently working on defining that term as we speak. But in short, it's a new term. It was coined in the mid-2010s by an ecologist named Dr. Mauro Galletti. He's a professor of ecology at São Paulo University in Brazil. And essentially what it refers to is the explicit study of the effects of animals on ecosystem processes like nutrient cycling, fire regime, and particularly of interest to me, carbon cycling. So why should we care about zoogeochemistry? And moreover, why should we study it? Surely we have enough ecologists studying animals, their behavior, their physiology, and enough studying ecosystem functions and processes like climate change and carbon cycling. And that is most certainly true. But what is also true are distinctly modern crises like biodiversity loss at the local and global scales. Human activities have resulted in unprecedented modern rates of wildlife loss. For example, terrestrial vertebrates like the zebra and the elephant that you see here, terrestrial vertebrate species worldwide have experienced a decline in abundance of about 25% since 1500 on average. And what's more is those declines are not occurring evenly across taxa. Generally, the larger the body size, the greater the risk of decline or extinction, given the relative needs of those animals. They tend to consume more food, they require a much larger home range, they reproduce at slower rates than smaller animals like rodents, and they experience human-wildlife conflict more frequently. And the same ecosystem without an elephant or a similar mega-herbivore wouldn't be down just one species. The functional outcomes of that elephant's loss on the landscape are significant, thanks to the specific ways that elephants and other mega-herbivores interact with vegetation, the physical elements of the landscape, and other wildlife. And the pressures that cause mega-herbivores like elephants to decline in abundance or to disappear entirely are likely to also impact populations of other large wildlife too, and subsequently the loss of the functional roles that they play in those ecosystems. So what does this have to do with the carbon cycle? A quick reminder of how carbon cycles throughout ecosystems generally is that carbon is stored in and cycles between different chemical forms in different stocks or pools, in the soil, in plant and animal biomass, and in the atmosphere as greenhouse gases like carbon dioxide. Plants extract carbon dioxide from the atmosphere via photosynthesis, transforming it into plant biomass, which is then either consumed by animals or deposited into the soil pool through death and decomposition. Some of that carbon stays in the soil pool as minerally associated carbon, but some is transformed back into atmospheric carbon dioxide via the respiration of soil animals as they break down soil carbon for fuel. And big animals have really distinct and measurable impacts on ecosystem carbon, both direct and indirect. This paper is from a 2018 paper, or this figure is from a 2018 paper led by my boss at Yale, Dr. Oswald Schmitz. And I think it really beautifully describes the relationships between big animals and the other organisms in their ecosystem and the physical structures of their habitats. They directly impact ecosystem carbon by consuming vegetation. They trample and they compact the soil beneath their feet, presumably multiple feet. They also interact with other animals such that they influence the behavior and the physiology of those animals. So there's a whole lot of direct and indirect effects of big animals on ecosystem carbon. So there are a couple of really dramatic examples that I want to share, and this one in particular is a really evocative example of the kind of effects that big animals and changes to their community composition can have on a whole ecosystem's carbon cycle. So wildebeest, which is the animal that you see in front here with the curved horns, they're an East African antelope species, and they're famous for their yearly migration around Serengeti and Masai Mara national parks. And essentially what happens is they follow the greening up of the savanna as the wet season sets in after a long dry season, grazing as they travel. And like many grasslands, the Serengeti and the Masai Mara savannas are net sinks for atmospheric carbon, meaning they take up more atmospheric carbon dioxide through photosynthesis than they release back to the atmosphere through respiration and other pathways. In the mid-20th century, the wildebeest population in Tanzania and Kenya dropped precipitously from the hundreds of thousands to the hundreds, as you can see here at the start of this plot in the black circles. And this was due to an outbreak of a cattle-borne disease called rinderpest. This basically resulted in the functional extinction of the wildebeest population, which then resulted in an increase in above-ground plant biomass because the wildebeest weren't there to graze on it. And while you might think this would be a good thing in terms of carbon uptake, right? More plants taking up more carbon through photosynthesis. In reality, it resulted in what were once small-scale yearly fires that were normal to huge hot wildfires that had tons and tons and tons of above-ground plant biomass as fuel to burn hotter and longer. During this period of time, the savanna went from storing more carbon than it emitted to emitting more carbon than it stored, thanks to this shift in the fire regime, which was supported by that increase in above ground biomass for fuel. And what ended up happening was not only all of that above ground biomass burning up, but a lot of below ground carbon vaporizing as well. So when the rinderpest vaccine was introduced in the 1960s, the wildebeest population started to rebound. And today, the previous carbon cycle functionality and the overall carbon budget of the system were restored. So how can we detect animal effects on carbon cycling? It's not terribly easy to create an experiment involving animals as large as, and that range as widely as most vertebrates are and do. But what we can do is observe the outcomes of experimental or circumstantial changes to the community networks that describe an animal community. So for example, we might experimentally remove one or several species from a network and observe the outcomes on the rest of the community, on the ecosystem structure, and on the system's characteristic functions. So this brings me to my case study number 1, the Kenya long-term exposure experiment. So this experiment is located at Impala Research Center and Conservancy, which is a working cattle ranch and wildlife conservancy in Laikipia, Kenya, which is in the center of the country in East Africa. And the Kenya long-term exposure experiment is located at the bottom left, the southwest corner of Impala Research Center and Conservancy, and it's a large herbivore exposure and domestic cattle grazing experiment that's been running since 1995. The experiment involves these big 200 by 200 meter or 4 hectare size plots where different combinations of large herbivores are allowed depending on what kind of electric fencing is installed. So the questions that I'm most interested in, how does experimental change to the large herbivore community influence carbon dynamics, I address in 4 of its experimental plots. So the first one is wild herbivores only, which is accessible to all of the large herbivores except for to Impala's own herds of domestic cattle. Wild herbivores plus cattle, which is accessible to all the herbivores as well as to periodic cattle grazing that occurs about 4 times a year in each of those plots. So this is mimicking the kind of grazing pressure that the surrounding savanna experiences from pastoral cattle grazing. Cattle only, which excludes all of the wild large herbivores using electric fencing, but allows for that same cattle grazing periodically around 4 times a year. And total exclusion, which excludes all of the large vertebrate herbivores, wild and domestic. So the question here, how does change to the large consumer community influence savanna carbon storage and cycling? So I'm going to focus specifically on carbon cycling for this talk. And one of the methods with which I characterize carbon cycling is by monitoring soil carbon flux, or the rate at which carbon dioxide is emitted from the soil to the atmosphere as a result of microbial respiration, root respiration, and the physical movement of carbon dioxide from deep soil layers to the surface. And this is typically done with something called a chamber-based method. So this is where a chamber containing a CO2 sensor or being routed to a CO2 sensor using tubing and a fan is placed on the surface of the soil via a collar, which you might have previously installed in the soil, and a gasket between the two to create an airtight volume inside which you can monitor CO2 as it builds up. So in my case, I used a chamber with an infrared gas analyzer in it, and I'm able to take minutes-long observations of increasing carbon dioxide inside that closed volume on the soil surface and calculate the rate of that increase to estimate the net emissions of carbon dioxide from the soil to the atmosphere in units of mass per unit of time per unit of area. So this is what that looks like in practice, placing a chamber on a pre-planted collar such that it creates a sealed volume on the soil surface and monitoring the increase in carbon dioxide concentration inside that chamber over the course of a few minutes. So here are some results from some of that work. So this is from 2019, looking at data collected from plots from, so on the x-axis here we have herbivore experiment treatment on from most herbivores present to no herbivores present. So wildlife plus cattle, just wild herbivores, just cattle, and no wild herbivores whatsoever. And on the y-axis here, we have flux rate in micromoles per meter squared per second. So you'll notice that there's a lot of variation in these data. There is some sort of discernible pattern. It's possible that the presence of cattle in the wildlife and cattle and the cattle only plots is depressing our flux rates that we're observing here, but it's quite variable and it's a little bit hard to detect any real patterns. But when I break the data up into two groups within each treatment, things get a little bit more interesting. So in this plot, the data are exactly the same, but I've separated the flux observations into those taken on open soil patches and in yellow, and those taken on soil underneath tree canopies in green in each of those herbivore treatments. So you can see that there seems to be a pattern with higher soil carbon flux underneath those tree canopies, no matter what herbivore experiment we're in, except for in the no herbivores allowed area, where we have pretty equivalent levels of flux in the open soil matrix and under the tree canopy. So the reason I split this up into these different elemental features, these different structural elements, is that there are significant animal engineering effects on the sort of physical structure of the savanna. So here you can see sort of a representation of what the savanna looks like using data collected from Drs. Grace Charles and Judith Sitters. And this figure basically just shows a schematic breakdown of what the savanna looks like with the different community compositions. So over here in the far left, we have all the wild herbivores and cattle. And as we move counterclockwise, we reduce the functional groups of wild herbivore or of herbivore groups, right? So here we just have wild herbivores, here we just have cattle, and here we have no large vertebrate or herbivores at all. And as you can see, as we move from all herbivores totally open, no fencing, to fully exclosed from wild herbivores and from domestic herbivores, we have a greater amount and total coverage of tree canopy cover. And this is important because the only tree in this ecosystem is a tree called whistling thorn acacia, Acacia japanolobium, and it's a nitrogen fixer. So they tend to be really biogeochemically distinct from the surrounding soil. We also have differences in the number and distribution of termite mounds, another biogeochemically distinct feature in this savanna that's really, really important for carbon cycling. So we decided to really dial in on these distinct landscape features, these structural elements of the savanna, to see if they were different from each other in terms of soil carbon flux. So we chose to do this in just the two most extreme treatments to really see if we could pick up on a pattern. So again, on the left here, on the x-axis, you're going to see the treatment where all of the herbivores are allowed to graze, including cattle. And on the right, the treatment where it's fully fenced off, no large vertebrate herbivores are allowed. And again, on the y-axis here, we have flux rate. And here we can see a slightly more discernible pattern, right? So all of these data were taken in the middle of the day in 2019. And then we can see that in pink here, termite mounds, in yellow or open soil, and in green under tree canopies. And there doesn't seem to be a huge difference in the herbivore plot, but in the exclosure plot, we see sort of this stepwise decrease in soil carbon flux from termite mounds, where there's a lot of biological activity, a lot of below ground venting, a lot of channels through which carbon dioxide can travel, the open soil matrix, and then under the tree canopy. So a little bit different from what we observed before, but still an interesting pattern. But this kind of pattern is really exciting and it's super interesting, but I knew that we were missing really big windows of time that were really important to sample carbon flux in this ecosystem. The first one is after it rained. So this is a seasonally dry savanna ecosystem, pretty low levels of carbon flux from soil respiration generally, unless it rains, right? So this is a drought-adapted ecosystem that tends to perk up really quickly after rainfall, become really biologically active. However, it's also an ecosystem with 50% clay in the soil. So we were just physically unable to get up to our field site after it rained. So there was this whole sort of period after rainfall that's really biologically active, really ecologically important that we didn't have any data for. And then additionally, we weren't able to collect data at any time other than during the daytime hours. And this is for pure logistical reasons. Nighttime hours are when the big animals tend to come out. And it was not safe to go out into these huge 4-hectare treatments to take soil carbon flux data at night. So we realized that we needed to capture soil carbon flux at the scale of animal influence, including greater replication. It was really hard with one survey chamber to get to the number of collars that I needed to measure in a couple of days across this entire Kenya long-term exposure experiment. You know, I'm just one person hiking through literally miles of savanna with this survey chamber, and it was hard to get the kind of replication that we needed without getting really noisy data. We also needed our tools to be more robust to damage. We needed to be able to have them be highly replicated across this landscape, but also not have it be like a huge deal if a cow kicked one, if an elephant stepped on one, if a monkey stole one, right? Like this is a really sort of ecologically challenging place to put sampling instruments. We needed them to be, you know, larger spatial extent. These are huge 200 by 200 meter plots. And critically, we haven't been able to collect data except during the daytime hours. We needed to be able to have 24-hour soil carbon flux monitoring capacity. So all of this is adding up to something that you see written on this slide a lot, which is low cost. We needed to make something that was cheap and autonomous, such that we could have high sort of spatial resolution, temporal resolution, but also a large spatial extent. So this is where the flux spots come in. So my graduate school mentor, Dr. Kelly Kaler, and I collaborated to design, test, build, and deploy an array of 18 autonomously operating soil carbon flux chambers in the Kenya long-term exposure experiment in 2019. We worked with undergrads in ecology, engineering, and computer science to pull this off. And this is a photo of one of them deployed on a termite mound in Klee with my sunglasses for scale on the bottom left here. You can see there's all of this electronics in the lid, including a carbon sensor, a temperature sensor, a humidity sensor, and a pressure sensor, and a linear actuator that opens and closes the chamber according to a predetermined schedule. So these flux spots stay open, like you see here, for 55 minutes out of every hour. For the last 5 minutes of the hour, however, they close, creating that sealed volume on the surface of the soil and collect high frequency data on soil carbon emissions inside that closed chamber for about 5 minutes. At the end of 5 minutes, the chamber opens again, the flux spot goes back to sleep, and the chamber vents to the atmosphere before starting all over again 55 minutes later. On-mound is closed. So as you can see, these chambers are operating autonomously and simultaneously, such that we can have a mound-based, flux spot over here, and 20 meters away have a flux spot located underneath a tree or on an open soil patch, and they will be collecting data that is comparable in time. So we deployed groups of 3 flux spots, so little triplicates of open soil underneath tree canopy and on termite mounds in the open plot here and in the total exposure plot here with no wildlife or domestic grazers. So this map here just shows approximate locations within one of the Klee replicates here. So this is 400 meters by 600 meters, so very large. This map doesn't actually show the termite mound locations, but each red circle encompasses a triplet group. So one on a termite mound, one under a tree canopy, and one on open soil. So these data are from that deployment, which took place for 2 months over August and September in 2019. And you can see that there's still generally higher fluxes on the termite mounds in pink compared to on the tree canopies in green or in the open soil patches in yellow. And funnily enough, that higher flux in the exposure plot that we saw before doesn't really seem to be detected here with the data taken repeatedly over 24 hours, rather than the survey data taken only during the daytime hours. Okay, so this next plot is a little chaotic, so I'm going to walk through it first before throwing up the data. On top, you'll see that I've split up the plot into structural elements, so termite soil in pink, open soil in yellow, and under tree canopy in green. So for each element, I have the same axis as before, from the totally open plot to the total exposure plot, and then flux rate is on the y-axis as usual. And on the right here, our key actually shows us that we're going to be looking at flux rates from dawn to day, and then to dusk and to night. So we're splitting up our data through time. This is the first time that we've had data that we can actually look at temporal patterns in addition to spatial patterns. Okay, so bam, we can see a lot of data here, but now that we have our axes oriented, I'm just going to walk through it. We can see that it's fairly clear that, as before, our termite soils are consistently higher in flux rate than the other structural elements. But across all of our elements, so all 3 structural elements in the savanna, we can see a repeated pattern of slightly higher fluxes in the open plot compared to the herbivore exposure plot. So the open plot here, where all of the herbivores are allowed to come in and graze, versus the total exposure plot here, which is completely fenced off from large herbivores. So the biggest thing we noticed, however, and I've pointed it out with these extremely subtle red arrows, we noticed a significant and repeating trend in our flux data across all of the structural elements and in both herbivore treatments. Higher fluxes in the nighttime hours, here represented by the dark blue bar on the far right of each plot, than in the daytime hours in orange. So this is surprising if you do anything with soil carbon flux data. It was super consistent no matter how we analyze the data. This was the pattern that showed up, higher fluxes at night. However, most other data sets that are taken over 24 hours for soil carbon flux show higher fluxes during the day. And this is because higher temperatures generally mean that soil microbes and roots are more biologically active, they're respiring more, and they're releasing more CO2 to the atmosphere during nighttime hours when everything kind of biologically shuts down due to cold temperatures and a lack of sunlight. So our data set was showing us the exact opposite of that pattern, and we kind of freaked out. We were like, did we build these wrong? Are they measuring something real, or is this how we constructed the flux spot? However, most of the existing data sets on soil carbon flux have been collected in temperate ecosystems like forests here in New England. And in those systems, flux is positively correlated with air temperature. However, in this tropical savanna system, which is wildly understudied compared to temperate systems, we found that flux was instead positively correlated with moisture. So again, this is a seasonally dry grassland, and during the height of the day, air and soil are extremely dry and hot. And as a result, biological activity slows down significantly. When dusk falls, dew formation is super rapid, super significant, and it provides a big influx in terms of air and soil moisture. So this creates a daily drying and rewetting cycle where we hypothesize that the soil actually experiences a spike in biological activity after receiving moisture in the form of increased humidity and dew. And this kind of pattern has actually been observed in the few other tropical sites that have 24-hour flux data collection, but those are in tropical rainforests. But these are the only other data sets like this that are collected in tropical ecosystems. And this plot here just shows from dawn to day to dusk to night, sort of this U shape in average relative humidity. This data is actually from the flux spots collected from those same instruments with their onboard humidity sensors. So the takeaways from this case study is that zoogeochemical studies on carbon dynamics require really high replication. So that way we can get both high resolution in the spatial and temporal sense, and also large scale. Whoops. Tropical savanna systems behave significantly differently than temperate systems in regards to the temporal patterns of soil carbon flux. We observed higher fluxes during nighttime hours across all of our treatments and structural elements, the exact opposite pattern that we would have expected based on historical data from other systems. And soil carbon flux is most consistently influenced by time of day or the ambient conditions at time of day, which interacts significantly with the presence or absence of large wildlife and small-scale structural heterogeneity. So we're still teasing apart the patterns in our analysis of this data set. It turns out it's really complicated, but we do see that significant effect of higher fluxes at night, and it is significantly interacting with wildlife community and small-scale structural heterogeneity, all of which is really intimately tied together. So it's not just wildlife loss that can affect a system, right? So the system in Kenya allowed us to sort of have this pretend wildlife loss experiment where we could say, okay, if we lost mega herbivores from the system, like has happened in a lot of ecosystems that previously had mega herbivores and where they went extinct, this is might be what we would expect to see in terms of the ecosystem's response. However, there are other ways that animal communities change in the modern day, and that includes the addition of species. So all kinds of ecosystems all over the world, especially given how highly connected the world tends to be now through shipping pathways and air travel, they have instances of introduced animals to ecosystems, and that can have sort of similarly dramatic effects on animal community, resultant ecosystem function or ecosystem structure and then function. So this is going to bring me to my second case study, looking at the case of moose in Newfoundland, Canada. So moose were introduced to the island of Newfoundland from mainland Canada about 100 years ago for hunting, for sport hunting. So Newfoundland is a beautiful boreal forest ecosystem. It had other animals, deer, wolves, bears, but it didn't have moose, unlike the rest of Canada. And so moose were brought over, two breeding pairs of moose were brought over for hunting purposes around the same time that the wolf population was locally extirpated. So this meant that the moose did not have a native predator to keep the population down. And the population of moose just exploded. And today, Newfoundland has some of the highest densities, if not the highest density of moose per unit area anywhere in the world. There's moose everywhere. They're famous for it. The thing about moose is that they really love baby boreal tree seeds, or seedlings, excuse me. So normally, boreal trees are light limited, which means that when you have a totally closed canopy, you have these little baby boreal tree seedlings, balsam firs, birches, maple, and they're kind of just lying in wait on the forest floor until a disturbance opens up a gap in the canopy, allows light to reach the forest floor, and then those seedlings can compete with each other to grow up and fill that gap in the canopy. And this is sort of this dynamic process of disturbances happening, gaps in the canopy forming, and trees growing up to fill those gaps. The thing about moose is that, like I mentioned, they love those baby trees. So instead, what can happen is a gap will form in the canopy from a disturbance, maybe a fire, timber harvesting, or an insect outbreak. And rather than those trees getting the opportunity to grow and fill the gap in the canopy and close it, moose are going to come and feed and browse on those baby boreal tree seedlings, not allowing that gap in the canopy to sort of heal from disturbance, and instead creating what are called moose meadows. So creating this positive feedback loop where moose are coming back to these meadows repeatedly to browse on baby boreal tree seedlings because of their high nutrient content. So I was really interested in looking at the relationship between moose and boreal forest structure and resulting effects on boreal forest ecosystem function. And I did so, or I am doing so, in two national parks. So Terra Nova National Park on the east side of Newfoundland and Grossmore National Park on the west side. And for anyone who's unfamiliar, Newfoundland is an island off the coast of Canada. It's the easternmost point in North America. So these moose meadows, you might be thinking like Julie Andrews, Sound of Music, spinning around and singing songs. I sure was, and I was excited to work in meadows after doing my dissertation work in, Kenyan savannas, which are beautiful and wonderful, but also full of spines and spiders and all sorts of things that are prickly. It turns out these meadows are not as bucolic as you might expect. They're actually really hard to walk through. This is my research technician, Lauren, last summer, and my other colleague, Ermila, hiking our way through moose meadows formed from a historical insect outbreak. And then here is a picture of a moose exclosure that the Park Service put up in the 90s. They've got a whole network of moose exclosures set up around the parks that allow researchers like myself to come in and sort of hypothesize what the forest would look like if moose were no longer there and allowed it to regenerate. So we have a whole lot of data on how moose interact with the forest now. So this plot here shows Grossmore National Park, that the park on the west side of the island right here, Terra Nova National Park on the right. And we're just looking at moose browsing activity inside of the forest gaps versus in mature forest stands. And you can see that there's just a lot more moose activity happening in those gaps. So we were wondering, what is the relationship between not only the structure of the ecosystem, so gaps versus mature stands, but also the level of activity. Does the fact that moose are sort of importing nutrients in the form of feces and urine influence the carbon cycle in those gaps versus in the mature stands, in addition to the structural changes that they wrought on the boreal forest structure? So this involves, of course, taking more soil carbon emissions data. So this is a picture of me taking soil carbon flux data in a mature forest stand. And this is what that data look like across Grossmore National Park on the left again, and Terra Nova National Park on the right. Here we're looking at flux rate as usual on the y-axis. And on the x-axis here, we have fluxes collected in the moose exclosures, fluxes collected in the gaps in the forest canopy, and then fluxes collected in the mature stands. Once again, you see a lot of variability here, both across the two parks, as well as across the different treatments. And this is the kind of thing that we are going to try to get at this summer with our FluxBot 2.0. So we've made some changes to the FluxBots. They have a higher accuracy CO2 sensor with them. Installed in them. They have a longer battery life because it turns out we can't rely on solar power up in Newfoundland the way that we could on the equator. They're more weatherproof. They newly, thanks to our colleague Connor Pan, have remote data transmission capabilities via the cell network, and they're more replicable. They have more off-the-shelf components as opposed to a lot of homemade components that we made ourselves in 2019. So this is what they look like in theory, and this is what putting them together looks like in practice. So this was at our Airbnb outside the park last summer. This was a neighborhood cat who was fascinated by the process. But the new ones operate very similarly to the old ones. They're open for 55 minutes out of every hour, and then once an hour for 5 minutes, they close. They stay closed for 5 minutes, take data on CO2 as it builds up inside that closed chamber, opens up again, goes back to sleep. But before it does that, it connects to the cell network and remotely sends that raw data, that accumulation curve, to a Google Sheet where we can monitor them in real time. So last summer did not go as planned. So they didn't actually work last summer, at least not yet. So we were able to collaborate with some folks at Harvard Forest, and I was able to go back to my roots in Massachusetts, where I'm from, and hang out in the woods with my mom and my dog for a deployment at a long-term ecological research site in a temperate forest. So we deployed a network of FluxBots across a couple of different sites in central Massachusetts, and we were able to detect differences in soil carbon flux in a healthy hemlock forest on the left versus an unhealthy hemlock forest on the right that is suffering from a hemlock wasting disease, resulting in gaps in the canopy and a much more diverse understory. So the plan as of now is to come back to Newfoundland in 2024 in June and July and deploy an array of FluxBots across both national parks, inside of gaps in the canopy, inside of mature stands, and inside moose exclosures, so we can try to understand if there are any patterns in soil carbon emissions in the mature forest, in the disturbed forest, which is being visited more frequently by moose and has greater biological activity in terms of the large herbivores, and in these moose exclosures, which sort of mimic what the effects of sort of moose eradication would be on forest structure. And our goal is to try to detect specific patterns in soil carbon flux in space and time within those gaps, within mature forest stands, and within moose exclosures, and work with Parks Canada to try to figure out, is there some sort of balance between moose population density and the boreal forest's historic ability to take up more carbon dioxide than it emits, to be a carbon sink? So some overall takeaways from what I just talked about for about 40 minutes now is that zoogeochemistry is a growing subfield in ecology that's going to require a combination of methods from both ecosystems and community ecology. And in order to do that, it's going to require the development of novel and accessible tools that allow us to monitor ecosystem processes at both high resolution in space and time and in scale, right? We're talking about experimental sites like my sites in Newfoundland that span entire national parks and require us to hike deep into the forest or deep into the savanna, deploy tools, and be able to leave them there to collect data without us, so that way we can get enough data to detect patterns that are biologically relevant to the questions we're asking. And lastly, we know that changes to large-bodied animal communities, whether it's species removal or addition, have cascading effects on the structure of the ecosystems in which they're found, both in the tropical savanna in Kenya and in the boreal forest of Newfoundland. And those changes on physical structure have cascading influence on the function of the ecosystems on which they're found, right? So I want to wrap up by just acknowledging the many, science doesn't happen in a vacuum. None of it does. I just showed you a lot of data and photos that I took myself, but there were a ton of really amazing people involved in all of it, including labs at UC Santa Barbara, like Dr. Hilary Young, Joshua Schimmel, and Kelly Kaler, Truman Young, who started the Kenya long-term exposure experiment in 1999, my lab here at Yale, Dr. Oswald Schmitz, and my lab at Memorial University, Sean LaRue, as well as Impala folks like Dr. Duncan Kimoyu, and particularly my collaborators across non-ecology fields, including Connor Pan, Vatso Patel, Vincent Beninati, Spencer Fry, and Grace Lewin, and John Gortzman, who helped us set up the Massachusetts component of this project. And quickly, I want to thank my funding sources, the National Science Foundation, the Yale Center for Natural Carbon Capture, and Parks Canada. So with that extremely long list of acknowledgements, I'm gonna just thank you all again for having me here today.
Thank you for attending the webinar. To learn more about the CIRUS-4 portable photosynthesis system or the EGM5 portable CO2 gas analyzer, visit ppsystems.com.